Hello, everyone, and welcome to the first international meeting in basic biomedical sciences, where will be our fourth seminar of this series. We are so happy to make an announcement that now you can be the next person to present your research in this event. The only thing you need to do is send us an abstract of your research, obviously in basic biomedical science, and our committee will be able to evaluate it to choose the best proposals. We will be receive all the abstract to participate at our official email, encuentro-intercbb@unal.edu.co. And you can start this process now. We have the company of Amy Jo Montgomery from the University of Missouri. And today she will be helping us with the moderation of our conference and any question you may have. Good afternoon, Amy. Welcome again to the National University of Colombia. How are you today?
I'm doing well, thank you. I'm so happy to be back again with the National University of Colombia. So thank you for inviting me to participate today.
Sure. We are so happy you are doing well. And now we can begin. Tell us about the conference today.
So today we will be presented with the fascinating topic of color Doppler ultrasound in animal reproduction. We will learn how this technique can be used not only for the diagnosis, but also used to predict reproductive performance in animals, even including humans.
Seems that the conference is going to be so interesting. But Amy, I don't know how we can predict reproductive performance in animals using color Doppler ultrasound.
I know Dr. Mohammed, who is our expert today, will be able to answer all of these questions. But Jasser, something I do know is that we can use this technique to measure the amount of blood that reaches the corpus luteum in the ovaries. It seems that this amount of blood is proportionally related to the amount of progesterone produced by this gland. We know this all because of color Doppler ultrasound. Isn't that so fascinating?
Yes, indeed, it is very fascinating. Okay, this is the moment of the truth. Amy, can you please introduce us Dr. Mohammed and tell us about basically his academic achievements?
Of course. So Dr. Elmetwally is an associate professor of reproduction biology and a faculty member of the veterinary medicine in the Department of Theriogenology at Mansoura University in Egypt. In 2004, he received his master's at Mansoura University in the Department of Theriogenology, and in 2012, he completed his PhD at Hanover University of Veterinary Medicine in the Department of Reproductive Biology. His thesis is titled Clinical Applicability of Non-Invasive Doppler Ultrasonography in Small Ruminants Throughout Pregnancy. He established a color Doppler ultrasound imaging unit at Cornell University while he performed his first postdoctorate. He completed another postdoctorate at Texas A&M University in the lab of Dr. Fuller Baser, where he researched the effects of catecholamines on the secretion of interferon tau and expression of genes for synthesis of polyamines and apoptosis by ovine tropoderm. He is currently scheduled to begin a postdoctoral study at the University of California, Irvine in the Department of Obstetrics and Gynecology, where he will study catecholamines and the treatment of preeclampsia in pregnant women. But now I would like to remind all of the participants to fill out the participation format to obtain a final certificate. This can be obtained by participating in at least 80% of the total conference. I would also like to invite all the participants to submit any questions using our social media. These questions can be submitted in either Spanish or English. We will translate any Spanish questions for Dr. Mohammed. Now I would like to welcome Dr. Mohammed to the National University of Columbia. We are eager to learn about this interesting topic. Dr. Mohammed, we're now ready for your conference.
Thank you so much, Amy, for this nice introduction. I am proud that I will talk today at the National University of Columbia about the application of color Doppler ultrasonography in animal reproduction. I know Yasser since long time ago, and we did a lot of research related to either the pre-implantation in experimental model sheep or in the color Doppler ultrasonography imaging in dairy cows and buffaloes and goats. Today, I will present some knowledge about the basic of color Doppler imaging application in animal reproduction, in addition to introduce some highlight about the futurism of color Doppler imaging using in reproduction in general. Today, I will say something about the background of color Doppler ultrasonography. Can be used to follow up the physiologic or pathologic state of either female or male genital tract. Color Doppler ultrasonography developed basically in human reproduction, and literally about from 1990 to begin to be applied in the animal reproduction. Doppler means Christian Doppler. Many people didn't know what is Doppler means. Doppler is the name of the scientist Christian Doppler from Austria. He worked firstly in astronomy. You know, the astronomy depends on the movement of planets in the sky. And if we compare between the movement of planets in the sky and the application of color Doppler in soft tissue, we will find a similar thing, which is the movement object. The movement object within the human or animal body is the blood. The circularity of RPCs is the basic thing that will depend on the application or reading of Doppler. The basic application of Doppler ultrasound is mainly depend on an effect known as a Doppler effect. In this slide, you will see the Christian Doppler from 18.3 to 1853 and his house in Austria. The aim of the present presentation is to verify at the end that the color Doppler ultrasound imaging is non-invasive technique would be successfully used to follow up the physiologic and pathologic state of male and male genitalia. In this presentation, we will go to highlight about some uses of color Doppler imaging, either for uterine blood flow, ovarian blood flow, and for the first time in this presentation, we will present something about the use of color Doppler imaging to follow up the changes in the vaginal blood flow. The basis of Doppler sonography at the first, as we said before, depend on the movement object. The movement object within the human or animal body is the blood cells. The ultrasound wave which transmitted from the machine to the cell, we can see it like F1. And this F1 should be reflected again to machine to have a wave. The difference between the frequency of the wave to the target blood vessel and the return back or rebound of the wave to the machine usually known as frequency shift or Doppler shift. If you're going to use the Doppler machine imaging, you may find something, the wave either under the line, we say this is the zero line. So you may find the wave under the zero line, or you may find the wave above the zero line. Anyway, there is no difference between the wave either down zero line or up to zero line, because all of the Doppler machine having a key that or a button that you can convert the wave from under the zero line to over the zero line. So from this slide, I want to highlight about the direction of Doppler waves. The Doppler waves may be either down zero line or upper to zero line, and this is usually according to the direction of the probe and direction of the blood within the vessels. If we're going to use the Doppler machine, we usually use the ultrasound probe. As you see in this charts, the waves which are transmitted from piezoelectric crystals within the ultrasound probe to the convex surface of the RBCs known as F1 or frequency number 1. And the reflected wave, we can say it like frequency number 2. The difference between frequency number 1 and the frequency number 2 is what give us the Doppler waves, and we say this is the Doppler effect. This is the Doppler effect. According to the frequency, when you have a look to this image, you will find something which interesting, and this is the distance or the angle between F1 and F2. We usually say, oh, this is the Doppler angle. This is the Doppler angle. Some of the Doppler indices will depend on this angle, and some other indices does not depend on this angle. And so we have to say something about the Doppler index or the Doppler indices, that these Doppler indices may be either affected by the angle or not. And so if you're going to establish research, you have to put in your mind which indices would be affected by the angle and which indices would be not. If you see in the first box, you will find the BFV, which is the blood flow volume, tau, which is time average maximum velocity, RE, which is the resistance index, BE, which is the pulsatility index, S by D, which is systolic by diastolic, BV, which is the peak velocity, and acceleration, the last one. As you see, most of these parameters usually developed firstly to use for human being, and then afterwards, since the early 19th, this indices used in the animal reproduction. So for these Doppler indices, we can divide these Doppler indices according to the effect of angle and the diameter of blood vessels into either qualitative assessment. The qualitative assessment in general depends on the presence or absence of in the diastolic blood in relation to the previous and the next systolic blood flow. From these sentences, we have to know that the Doppler indices usually depend on systolic and diastolic blood flow, and we have in or maximum systolic and maximum diastolic. According to the systolic and the diastolic blood flow, we usually develop or calculate the Doppler indices. In the quantitative, it is usually depend on the description. in the description of wave, and I will show you how we qualify the Doppler wave now. The wave characteristic either above the zero line or baseline or under the zero line. Increase in diastolic or decrease in diastolic, either for increase in systolic or decrease in systolic blood perfusion. These changes usually, through these changes, we can differentiate between the physiological and the pathological state of the male or male genitalia. Semi-quantitative ANSYS or semi-quantitative Doppler ANSYS. The semi-quantitative Doppler ANSYS depend mainly on the description of blood flow ANSYS which are independent of insunation angle or Doppler angle. I have to stop at this point because any time we qualify or quantify the Doppler ANSYS, there is some parameters that with minimum change of insunation angle or Doppler angle, this usually leads to increased change in the parameter which we read. And so most of the Doppler paper or publication or studies depend on both. You have to use both. The parameters which depend on the angle and the parameter which does not depend on the insunation angle. The semi-quantitative parameters which does not affect the insunation angle is the PE polystability index, RE resistance index, peak velocity, and time averaged maximum velocity. So when you're going to study any Doppler effect for any condition, you have to use either 1, 2, or 3 from these ANSYS. So if you're going to read most of the Doppler publication, you will find PE, RE, time average maximum velocity, or 1 or 2 of them, in addition to the other parameters which are affected by insunation angle. We can say at the same time the insunation angle as well as the diameter of blood vessels both affect the reading of the Doppler ANSYS, which depends on the angle and blood vessel diameter. The semi-quantitative parameters in general provide important information for the researcher and the clinician to understand the extent of vascular perfusion. For example, the peak systolic velocity, time average maximum velocity, both have a negative relationship with the BE and the RE. And this means that the relationship between the polystability index and the resistance index is positive relationship. So there is a positive correlation between BI and RI, and there is a negative correlation between either BI or RI with the peak velocity and the time average maximum velocity. Quantitative Doppler parameter. The quantitative Doppler parameters means mainly the blood flow volume. The quantitative blood flow Doppler parameters depends mainly on both Doppler angle and the diameter of blood vessels. This means that with increasing of either the diameter of blood vessel or the Doppler angle, this means the increasing of the blood flow volume of the target vessel or organ. If we're going to see here the Doppler assessment, for example, in these 3 Doppler waves, if we take the third one, we will find, for example, presence of peak systolic and absence of end-diastolic velocity. At the middle one, we will find the peak systolic and end diastolic. In the first one, we will find the peak systolic and end diastolic with presence of notch. The presence of notch is a descriptive, so this is a qualitative. Assessment for the Doppler wave. The distance between the zero line or the baseline and the end diastolic, we can put this also in consideration to quantify the assessment of the Doppler waves. This is for example. And the third one, for example, the absence of end diastolic velocity, we find this in sheep and goat until week 12. Also in the human being, until week 12 of pregnancy, there is no end diastolic velocity. So the mean blood vascularity depends mainly at the first on the systolic and not the diastolic. In the Doppler assessment, in the machine of all color Doppler, we usually can manage this assessment of parameters. In the machine software, you can use the machine software to adjust Z. From the left panel, PS peak systolic, end diastolic, mean diastolic, time averaged maximum velocity, BI pulsatility index, resistance index, peak systolic by end diastolic by peak systolic, acceleration, time averaged mean velocity, and also heart rate. What I want to say here that according your adjustment to the parameter within the machine, you can find the parameter you have to use in your research. In most of publication, you will find one or two or three from these parameters. In addition to that, the blood flow volume. Why in this panel we didn't find the blood flow volume? I cannot have the blood flow volume except when I adjust the intonation angle or Doppler angle and the diameter of blood vessels. And this is usually adjusted manually. The adjustment of the intonation angle, as you see in this image, and the diameter of the blood vessels is usually manually adjusted. What does it mean? This means that if I going to see the blood flow volume, for example, for this Doppler imaging, I can say, for example, the Doppler angle is 30 or 40. Someone can adjust this angle to 20 or 30. The other is using the measurement to measure the diameter of blood vessels. But in this situation, as I will show you in videos now, we usually take from 3 to 5 measurements and then take the average of these measurements to say, oh, the diameter is blah, 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 and the angle is blah, 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 and then according to introducing of these two parameters, you can have the blood flow volume. So, in all panels for Doppler management, you cannot find the blood flow volume. Why? Because you have to introduce manually the angle and the diameter of blood vessels. Anyway, we can choose 3 from this. We can take peak systolic, time average maximum velocity, pulsatility index, resistance index. We can take time average maximum velocity, BI, RI, systolic, diastolic, which is known as resistance impedance. If you're going through all Doppler literatures, you will find the three of these: time average maximum velocity, PARE, peak systolic velocity, BARE, maximum velocity, BARE, or the resistance impedance. Resistance impedance means the systolic by diastolic. We published recently something about the acceleration of the blood within the blood vessels according to the stage of pregnancy, and we can show this later. I want to say something here. How can I locate the blood vessels? The main basic idea to use the color Doppler ultrasonography. What does it mean? This means that if you're going to take the Doppler imaging for the carotid, for example, the carotid artery, or the femoral artery, or the aorta, or the external iliac artery, internal iliac artery, it's all having movement object, which is blood. So the basic idea in using of the color Doppler imaging is how to locate the blood vessel, And for us as a veterinarian, we have a concern in reproduction with the blood vessels or the blood supply for the female genitalia. The main blood supply for the female genitalia is the middle uterine artery. When we are going together to see the origin of the middle uterine artery in cattle and similarly in buffalo, we can find that from the abdominal aorta there is a branch which is the internal iliac and the other extended at external iliac. Well, I will show you now how I can locate the internal iliac artery from the abdominal aorta. And from the internal iliac artery, there is umbilical, and from umbilical, there is a middle uterine artery. Please see with me in this. You will find here the artery which go down is the external iliac. So the external iliac usually go away. So basically, I usually try to locate the abdominal aorta and then go down in the direction of the female genitalia. I will find that the external iliac artery go away, and then the internal iliac artery give me the umbilical artery, and from umbilical artery is the middle uterine artery. In the middle uterine artery, I try to adjust the window. This is known as the Doppler window. What does it mean? This means that only the color imaging will be present within this window. So I have firstly to locate the blood vessels and then to adjust the angle or the window that I put the blood vessels in between this window. And according to the video you see, I have to have from 3 to 5 similar waves. If the Doppler waves are dissimilar, I cannot use it in measurement. When I have 3 to 5 waves, similar waves, I take these waves for my measurements. And then, as you see, I will take the diameter of blood vessels from 3 to 5 times to have mean diameter. You see here how the external area go away and the internal area have the umbilical, and from umbilical in the middle uterine artery. This nice sketch is from Daniela from the National University of Columbia. She helped me to do this graphic. This graphic, for example, illustrates the blood flow to the female genitalia in general during pregnancy. And if we're going here in this sketch, we will find the aorta. The aorta at the level of the pelvic cavity or the pelvic entrance is usually continue to give the external iliac, and here is the internal iliac. From the internal iliac, there is umbilical, and then there is the middle uterine artery. The middle uterine artery usually gives two branches, the cranial branch and the caudal branch. In general, we did a lot of work to see if we put the ultrasound probe either near to the bifurcation of the middle uterine artery or away from the bifurcation and see if this affected the Doppler parameter or not, and we didn't find there is an effect for the side of the middle uterine artery on the parameter that we have. The middle uterine artery gives the cranial and caudal branch, and then there is a placentons. We have also for the first time described the blood perfusion for the placenton during the whole gestation period. I will show you this later on. And there's the external iliac artery and vein. And then when you saw here, the internal iliac usually give a branch after about 25 centimeters, which we say the vaginal artery. And we did for the first time an study to evaluate the changes in the vaginal artery during a pregnancy. This is anatomy. Whenever you put the ultrasound probe in arterial blood, you will have the same wave. Again, whenever you put the ultrasound probe in any artery, you will have the same wave. But the first and last thing in all Doppler study is the description of how to locate the blood vessel. For example, here is the endometrium for the cows. And as you see, according to this dots, this describes how is the blood perfusion to the endometrium during pregnancy. As you know, in the last decades, in human being, in vitro fertilization in general, they depend firstly on the thickness of endometrium before they transfer the embryos for women. But nowadays, they depend mainly on the blood perfusion for endometrium, whatever the thickness is. So we can see that the blood perfusion for the endometrium play the master or the most important role for fixation of the transferred embryos. Here, for example, is the imaging, and this green window, I say this is the window, Doppler window. According to these dots, we can say the blood perfusion is increased or decreased according to the density. Here, for example, the endometrial blood perfusion for the endometrium. We have to see this image before transferring of embryo, for example, either in human being or in animal, whatever the thickness is, because there may be the thickness of the uterus may be thin, but the blood perfusion is so bad. And how the embryo can be fixed within the uterus with bad uterine blood perfusion or endometrial blood perfusion. We use Doppler also to follow up the healthy status for the embryo within the uterus. For example, here is the intrauterine fetal circulation. We all know that the intrauterine fetal circulation is reversed to the normal circulation after birth. This circulation begins with the umbilical arteries or umbilical vessels, and again, for example, to have or to evaluate the umbilical blood perfusion in cattle and sheep and goat, for example, you have to locate 4 vessels, 2 arteries and 2 veins. But in male and in human being, you have to locate only 3 vessels. The blood is usually come from the uterine, the umbilical arteries to the ductus venosus. I will tell you now how to locate the ductus venosus within the fetus, and then to the heart, and then the blood is distributed. So if I'm going to study the intrauterine circulation, I will study the changes in the umbilical blood flow, changes in the ductus venosus, changes in the aorta blood perfusion. So I can describe, or for example, by the end, I can say if this fetus have to continue until the end of pregnancy, or I have to terminate this pregnancy. Also, this may indicate the health status or the development of the fetus within the uterus. And most of studies now all over the world concerned with the IUGR fetuses. The IUGR fetuses, I can differentiate if this fetus is IUGR fetuses or not, according to the umbilical and blood perfusion from the aorta. This is, for example, how we can use the Doppler ultrasonography to study the internal fetal circulation. There are many Doppler machines all over the world. We may have Sonosite, we may have GE, we may have Toshiba, according to the place. But by the end, we have a probe, a Doppler probe. The Doppler probe depends mainly on the piezoelectric crystals which transmit and receive the ultrasound waves. If we're going to study, to have in study on the ovary, we may say the ovary has two main structures. The ovary have a follicle and the CL. I want to stop here, for example. If you see for the first time this ovary, you will say, for example, this ovary have only a follicle. There is no evidence for the density of the CL, as we all know about using the normal beam mode ultrasound during the examination. I cannot see here a corpus luteum. When I'm using the color Doppler ultrasound machine, I can say, for example, here is the follicle, and I can take, this is taboo for the Doppler imaging of ovary. In all ovaries, I can find follicle and find CL. Here is the early CL. This is early CL, so the density of the tissue it not looks like the normal density of the tissue. Here is the CL, and here will be second CL. And as you all know, there is a relationship between the follicle and the CL, like Tom and Jerry. As the development of CL means is the regression of follicle and the vice versa. After imaging of the ovary, I can expect how the endometrium looks like. If the ovary has a well-developed corpus luteum, this means that the blood perfusion for the endometrium is well. And this means that, or I can predict that the conception rate for these cows will be high. You see here the imaging the beam mode and then the color Doppler imaging. You have to see first the beam mode, and then you convert your machine from the beam mode to Doppler mode. Here examination for the ovary, for example, you can see here the density of CL, as we all see in the beam mode imaging. But with this window, I can see this is a Doppler window. We can see the perfusion of the CL. You can predict if this cow going to be pregnant or not from the blood perfusion of CL. If the CL is active. I can say this is active CL. Why is active CL? Because there is a huge amount of blood within this corpus luteum. I can say if I see this corpus luteum after insemination or at the time of the prostaglandin F2 alpha shot, this animal would be pregnant. Okay, let's see here before imaging. You will find the echogenicity, the granular echogenicity for the corpus luteum. But here is something that we say the corpus luteum cyst. The main question here is the corpus luteum cyst affect the blood perfusion for the corpus luteum or not? We did an experiment when I was in Hanover and we didn't find any effect. We didn't find any effect for the presence of the cavity and the blood perfusion for the CL. So we usually depend on the blood perfusion, not depend on the size of cavity within the corpus luteum to predicate how is this corpus luteum active or not. After imaging of the corpus luteum by the color Doppler or power Doppler imaging, you have to measure the blood perfusion for this corpus luteum. And here you have two ways to use. You may either buy a software which is a pixel flux for measuring of the blood perfusion, either for uterine or for ovarian, or you may use this image with a free program which is the ImageG program, or within the field, because when I was at Cornell, we had a hypothesis that the Doppler machine is very important for every vet because Doppler machine imaging equal money. I will describe this later on. So we may not depend on either pixel flux or ImageG. We can say, for example, to observer, or according to the multiple observation of the person who did the color Doppler imaging, I can say without any measurement, this is a good blood perfusion, this is a bad blood perfusion, according to the amount of the blood in the contour of the corpus luteum. This is, for example, the pixel flux from the Chameleon software, which is present in Germany. And if we don't have to buy this software, we can use the image which is totally free by internet. I will take some examples for examination of the ovarian blood flow in cow, either in pregnancy or in cycle. In case of pregnancy, in case of the pregnancy or in cycle, for example, at the first days in estrus, this is the days in estrus in the X line, and here in Y there is 2, the mean diameter of the CL, and here the number of color pixels, which measured by either ImageG or the pixel flux. If you see here, for example, at day 7, 9, 11, 13, you will find increase in diameter, and with increasing of the diameter, the pixels also increase. The pixels of the blood perfusion or the blood perfusion for the CL is increased. After day 16, there is usually decreasing in the blood perfusion if this cow does not come pregnant. Again, for estrus cow, we usually say that the estrus cycle is the time between two ovulation. So I have to catch the CL at certain time to have my management and reproductive program. For example, the GBG program used for the estrus synchronization of Theriogenology. As you see here, at the time of beginning, there is ovulation. For this ovulated follicle, the cells become lutein cell to be mature CL at about 16 or 17. The maturity begins after ovulation, and the CL become mature at about 12 days after ovulation. From 12 to 16, the plateau of the CL size or blood perfusion or the serum progesterone become the same. After 16 days, the CL may either gravitate to be pregnant female or cyclic, and so have to be died by the prostaglandin F2 alpha secreted from the endometrium. And then there is another follicular wave and so on. I have to save money at this point, because if I'm going to use the GPG in my reproductive managemental program, so I have to give shots for prostaglandin F2 alpha at the maturity of CL. It may be either at day 9, day 10, 11, 12, 13. Most of programs depend on the 11th day, because the conception rate will be higher and the CL will be mature, and at this point affected by the exogenous prostaglandin F2 alpha. The luteal blood flow in cyclic, non-pregnant, and the pregnant cow. If you see here in this diagram or figure, you will find this green is the green is the pregnant. So with the pregnancy, the blood perfusion to the CL is increased. So, for example, if I take color Doppler imaging at day 15 or 16 from the cycle, I can predict if this female will become pregnant or not from the blood perfusion. At this day, as you see here, the blood perfusion is higher in case of pregnant than cyclic and non-pregnant animal. This is the effect of the human choriogonadotropin in the luteal blood flow, and as you see here, the relationship between the area and the luteal blood flow and the plasma progesterone. We found by the end that there is a positive correlation between luteal blood flow and the plasma progesterone, and not the luteal size and the plasma progesterone. This is the GPG program, and so here is the luteal blood flow for the follicular and luteal. The effect of shortened diestrus, this is, I will say something about this later on, about the use of the prostaglandin F2 alpha exogenous at this point. At this point, I have to confirm that the CL is mature, and I will say, I will show you how to differentiate between mature and the immature corpus luteum later on. And here, for example, the follicular blood flow and follicular size and the luteal blood flow at the time of prostaglandin F2 alpha in different waves. Here, for example, we use the color Doppler for pregnancy. If you see here the heartbeat from the fetus, you say, oh, this is a positive pregnant animal. When I was at Cornell, I asked the professor there, when you use a Doppler, they say we use the Doppler to see if the fetus is live or not. Live or not by normal ultrasonography means the presence of heartbeat. But here you can see the blood within the heart of the fetus during early pregnancy. Like that. This is a very nice video showing how we can use Doppler for early pregnancy diagnosis. Color Doppler imaging for the umbilical blood vessels, I have to go fastly. I'm sorry. Here, for example, you have to locate the 4 vessels at 1, 2, 3, 4, 2 arteries and 2 veins. This is the umbilical blood, the umbilical cord in case of ruminant. Cattle, buffalo, sheep, goat have 4 vessels. I have to locate these 4 vessels and then quantify the changes in the blood. This is, for example, to how to locate the blood vessels using the Doppler 1, 2, 3, 4. I locate the vessels and then take the Doppler wave to evaluate the health status of the fetus within the uterus. Here, for example, how to locate how the blood comes from the umbilical blood vessels to the ductus. If you're going to do any study about the intrauterine fecal circulation, you have to locate the ductus venosus within the liver. And this is how it located during the Doppler imaging. In Cornell, we usually say use of color Doppler equal money, because during the managemental reproductive program, I have to give shots from prostaglandin F2 alpha. If I give prostaglandin F2 alpha and the corpus luteum is large and not mature or not active, this means that I did nothing. So I have to use color Doppler machine to say if this cell will respond to my external shot from prostaglandin F2 alpha or not. And this is the basic idea for our project, that every vet should have a color Doppler machine during the examination within the field. And this is the aim of the study, and this is the design. And this is, for example, the preliminary results for this study. We see here now how the active CL is located. We saw the blood perfusion from all contour of blood vessels. In experimental design, I have to begin imaging of the ovary either by color Doppler or beam mode at the shot of GnRH, at shot of prostaglandin F2 alpha, at shot of GnRH, at time artificial insemination, and after time artificial insemination. I show you before, at the time of prostaglandin F2 alpha, if there is activity or active cell, I can give the shot at this time. Otherwise, I cannot. I don't have to give the shot because it's nothing. I lost money. I lost money at this point. Also, after at the time of the GnRH, we can say that this female may become pregnant or not. And this is, for example, the luteal size and the luteal blood flow. At the end, we have one thing to say, that the activity of CL in general does not depend on this size, but depend on the luteal blood perfusion. And this is the preliminary result for this study, indicating that when I begin the Doppler imaging, when I begin the managemental program, at the corpus luteum present at the first GnRH shot, the luteal blood perfusion will be high, and the evidence of pregnancy or the incidence of pregnancy also will be high. This is the luteal blood flow. If there is a CL at the first shot of GnRH, the luteal blood flow at the shot of prostaglandin alpha will be high, and vice versa. And here also the conclusion for this, either for progesterone, corpus luteum tissue size, and corpus luteum blood perfusion. We find by the end that the serum progesterone is highly correlated with the blood perfusion than the size of CL. This is active CL, and this is inactive CL, although it is larger in size than the previous one. But I see nothing here in the Doppler window. I cannot give the shot at this time. Here, for example, if we have 2 or 3 persons for evaluation or from experience in the field, I don't have to go to my PC to measure the blood perfusion. I have only to see and say, this is active CL, this is inactive CL. And so I have to decide to give the shot or not under the condition. This is results for this study and the analysis, and we will go to publish this study soon. If I'm going to see some of the artifacts for the Doppler imaging, for example, here there is a multiple artifact in the, this means that there may be some fecal matter, the rectum is not clean, there is something within the probe, I can see this at some times. Is aliasing. The aliasing means that there is a cut in the Doppler wave. A half become above the zero line and the other half become under the zero line. So I have to adjust the blood vessel and the examination. Here in the mare, for example, the scoring for the mesometrium and endometrium. Here high score because there is multiple dots from the Doppler imaging and power imaging, and here is minimum scores. Here also for the follicle in mare, here how to locate the blood vessels in mare for examination, either during the cycle or during the embryo transfer, because the blood perfusion for the uterus play an important role during the embryo transfer or success of embryo transfer process in mare. Here is the corpus luteum blood perfusion in mare. It looks like in cattle, and here is the program for the pixel flows. The corpus luteum in pregnant and non-pregnant mare. In pregnant mare, I found that there is high blood perfusion for corpus luteum than non-pregnant mare. The same is here. Either for plasma progesterone, we say that the CL blood perfusion have a direct relationship with the blood progesterone than the size. And here is the same. At the end, for this section, we can say the luteal blood flow is more appropriate indicator for luteal function during bovine or even in equine cycle than the luteal size. One for the futurism of using of the color Doppler imaging in the veterinary science or veterinary reproduction is how to locate the vaginal blood flow. From this image or from this sketch, we can see that the distance between the uterine artery and the vaginal artery is about 25. We have measured this. We had measured this distance before. We have a cadaver. We have a staining for the blood vessels, and then we measure the distance between the two arteries. And so we finally know as the distance from the middle uterine artery to the vaginal is about 25. And here, how the vaginal artery looks like during the rectal examination. This is away from the vaginal arteries, and this is a nice video for one of our study relating to how to locate the vaginal blood, perfusion transrectally. You see, it looks so nice. And the distance from here to the beginning over here, we can find the middle uterine artery. This is around 25 centimeters. And there is many study will become soon related to the evaluation of vaginal blood flow, because we know that the vaginal blood perfusion play an important role in perfusion of the gravid uterus during the late gestation. Also, the vaginal blood perfusion play an important role during estrus, during copulation. We can study, we already have this study, and it will be published soon, about the effect of either the position of semen or copulation, normal mounting on the vaginal blood perfusion. We can say that nothing equal to the normal copulation in case of female, because we find that the blood perfusion during the copulation is more than artificial insemination with a native ejaculate, is more than the same application of seminal plasma or either sperm within the uterus. This is a very interesting study. And so this is the first time that we describe how to locate the vaginal blood perfusion in case of cattle and buffalo, for example. And here is the first publication that describe the blood perfusion, the vaginal blood perfusion during the pregnancy in case of buffalo, for example. And also this is the first study to study the placental blood perfusion during the pregnancy also in buffalo. We also had a PhD student who work now in relation to describe or to study the changes in the vaginal blood flow during the prepartum period, the drying period, and the early postpartum period, and the results are so interesting and so excited. This is the difference between the Doppler wave for the uterine and the vaginal artery. Here, for example, this is the placental blood perfusion. We can say that by our colleagues, the blood perfusion in the normal cows is more than the blood perfusions for placenta in case of retain. We are running a study to study the difference in the placental blood perfusion in case of the cow which will having retain the placenta and the other cows in which the placenta will come loosely. We find an interesting results related to the engorgement of the blood within the feto-maternal section, which is the placentome, and the blood perfusion from the placenta. And this publication also will come soon. It is so interesting. This is the changes within the placentome during pregnancy, and by our colleague in clinical pathology, we can find a correlation, or we did a correlation between the uterine and the vaginal blood perfusion during pregnancy. Not only that, but we did a correlation with the biochemicals, like the total lipid cholesterol triglyceride. And we had a negative correlation, increase of total lipid cholesterol triglyceride. This means decreasing of blood perfusion either for the vagina or uterine. And this is the result for first time describe this relationship. And this we are so excited from these results. We have running a project now around the use of the color Doppler imaging in uterine torsion, and we found how the uterine torsion affected the blood perfusion for the uterus, either in case of cow and the buffalo. And here in torsion, for example, there is nothing in relation to the placental blood perfusion, nothing in placentome. This means how the twisting of the blood vessel affected the blood perfusion to the whole uterus compartment. We have an accepted paper recently about the use of the color Doppler imaging to measure the fetal heart rate, gestational length, and fetal birth in case of a stressed animal. We all know now the whole world is under stress, for example, from this COVID. Can we say that the people who are stressed or have a panic from the disease or any other condition, the uterine blood perfusion or the intrauterine fetal development is the same like in the normal one or not? We published for the first time here in Animal Reproduction the effect of the stress on the fetal birth weight and length of gestation in goat. And here is our interesting results is that the length of gestation or the gestational length in a stressed animal is usually decreased than the normal animal. And also the fetal weight decreased in relation to the fetal weight in normal non-stressed animal. But no one can know why the heart rate either in a stressed animal or non-stressed animal is the same. We didn't find any difference in the heart rate. There may be something related to the nervous accommodation or the nervous effect on controlling of the heart rate from the fetus. Also, we're running a study about the effect of mounting, mating, artificial insemination, and intravaginal deposition of raw semen or seminal plasma on the vaginal and uterine blood flow in the German Holstein cows. Here is how looks the Doppler waves for the vaginal and here how looks for the uterine artery. And here is the result. We didn't find any of our treatment is like the normal amount, the blood perfusion until 12 hours from mounting, increase in the uterine and also in the vaginal blood perfusion. And this was interesting. I would like to thank all of my colleagues in addition to Professor Heiner Bovine from Germany, and also my mentor in PhD, Sabina Meineke-Tillman, and her husband, Bukar Meineke, for their help me during my residence in Germany. And thank you for attending this presentation. And I am ready now for any question.
Dr. Mohammed, thank you so much for your intriguing presentation and the pivotal information that will improve performance in animal reproduction. Now let's take a look at some questions. Our first question we have looks like we are wondering how early in a cow's pregnancy can we detect the umbilical blood flow?
So during the early pregnancy in cow, it would be after around 40 or 50 days, because the heartbeat begin at day 25 of pregnancy. And so from later on, from 30 or 35 day, I can manage the umbilical blood flow in case of cattle. And here we have to notice something which interesting. The pregnancy in cow have two stages, the ascending and descending. So far the fetus is present in the pelvic cavity. I can see the umbilical blood perfusion. So I can examine the umbilical blood perfusion from day 40 until about 5 months of pregnancy. After that time, I cannot examine the umbilical blood perfusion. Why? Because the fetus comes down within.
Wow, very fascinating. Okay. So we have another question here. We're wondering, how can we distinguish any arteries from veins on a color Doppler ultrasound projection?
This is an interesting question. You know, the difference between artery and the vein is the pulsation. And so, for example, if someone is going to be sick, he usually uses the radial pulsation to see if he is okay or not. And so, the pulsation of arteries is usually high. Why? Because the blood is pumped from the heart to the blood vessels. So, the systolic and diastolic is more distinguished in arterial than in venous blood. By the Doppler imaging, I see usually this difference between systolic and diastolic. When you have the vein within the window, you cannot find a difference.
Very good. So our next question that we have are the ultrasound waves that are passed via the probe or the sound waves that are bounced off the blood, are they harmful in any way to the animal?
Yasser, no te escuchamos. Desde que empezaste a hablar no te escuchamos.
Bueno, dale entonces otra vez. ¿Ahí me escuchan?
Dale.
Perfecto. Para todos los asistentes del primer encuentro académico internacional de ciencias básicas biomédicas, pues las condiciones de nuestras conexiones han estado un poco difíciles, como ustedes se pudieron dar cuenta. Hemos estado tratando de lidiar con estos problemas de conexión, pero hacen parte precisamente de nuestro trabajo remoto desde la virtualidad. Hemos recuperado al doctor Mohammed. Les recuerdo que él está desde el Cairo, Egipto, y estamos ubicados en Colombia, Estados Unidos y el Cairo, Egipto, así que ustedes se podrán dar cuenta de que la Universidad Nacional de Colombia hace grandes esfuerzos para mantener esta comunicación con ustedes, los asistentes. Yo voy a darle el paso otra vez a Emi Montgomery para que sigamos la dinámica de las preguntas, y al finalizar vamos a dar respuesta a algunas de las preguntas que ustedes, los participantes, tienen respecto a la dinámica del encuentro, y adicionalmente vamos a dar una información importante de cómo usted puede ser el siguiente investigador que presente en este espacio, en el primer encuentro académico internacional de ciencias básicas biomédicas. So vamos a ir otra vez con el Dr. Mohammed. So, Emi, go ahead with the third question you had for Dr. Mohammed.
Hello, Dr. Mohammed. It's nice to have you back. So our third question was, are the ultrasound waves that are passed via the probe or the sound waves that are bounced off the blood harmful in any way to the animal?
At all, no. Because as you know, Amy, nowadays many people use ultrasound to treat some of condition in human being, and there is many studies that already done to see if the waves or the frequency, the effect of frequency, either high or low frequency, may have a detrimental effect on the tissue, and they didn't found anything.
Wow, that's good. So we have another question here. What are the preparations that should be made for an animal before a Doppler ultrasound is used for reproductive evaluation?
So interesting question, Amy. It is very important question because, for example, I have to say, to examine one animal for Doppler imaging, you have to take from 20 to 30 minutes. Why 20 to 30 minutes? Because you all know if the animal just move his or her leg, this means that the movement of the blood vessels from the window of Doppler. And so sometimes for cattle, buffalo, for example, they may give the animal anesthetic agent, which is, we say, posterior epidural anesthesia, or some people study the effect of xylazine in the uterine and blood flow, and they didn't found nothing. So the animal have to be calm during the examination. I have to sedate the animal sometimes if the animal is vicious, and this is very important, you know, that you have to be patient during the Doppler examination, because you have to fix your animal very well. Otherwise, you will not have any Doppler waves.
Very good. So our next question is, in your opinion, why is Doppler ultrasound not more widely used in animal reproduction evaluation?
In my opinion, Amy, when I was at Cornell at 2014, we have a project there that says every vet in the United States should have an Doppler machine. So we want to do some studies to learn the people how by observations they can differentiate or can quantify the Doppler imaging. We can say, Amy, for example, the Doppler machine are expensive. You can say, for example, here in Egypt, to buy a Doppler machine, you have To buy, you have to have around $10,000. This is for the Doppler machine. On the other side, the other machine, the B mode, it's around from $2,000 to $3,000. So it is an expensive machine.
I see. So our next question is for embryo transfer. The corpus luteum assessment in a recipient is a really common test. But how can we differentiate a functional corpus luteum with a functional regression of corpus luteum using Doppler imaging?
For the recipient cows, I usually know very well the time because I have to prepare this recipient cow. For the regressed corpus luteum or inactive corpus luteum, there is nothing in the contour of CL. The blood perfusion in the contour of CL is so low. But when the CL is functioning, as you see in my presentation, you will find a highlight in all contour of CL.
I see. This makes sense. So we have our last question here. What are the most important factors that affect the color flow image during the bovine reproductive assessment?
Important factors, right?
Yes.
The important factors in general for color Doppler imaging, number one, you have to train yourself more and more to how to locate the blood vessel. I usually say that to locate any blood vessels, it's so easy, but the target vessel is so hard. You have to be professional and to train very well, for example, to do rectal palpation and to locate the different organ within the pelvic cavity. This is one. And so you have to be good practitioner, good trainer, well-trained person to use the Doppler machine. The second is to differentiate between which parameter you want to use as a researcher. You have to use some from that which affected by the inzonation angle and the diameter of blood vessels with other parameter that does not at all affected by inzonation angle or the diameter of blood vessels like resistance index and pulsatility index. So if you're going through the literatures, you will find a mix from these. But in some literatures, you found only the parameter that does not affected by inzonation angle or the diameter of blood vessels. And so first you have to be well practitioner or well-trained person. Second, you have to decide which parameter you need. Third, you have to you have to train yourself about the Doppler imaging of different structures, because the Doppler imaging, for example, for the CL is different than the gradient follicle, and so anatomically you have to differentiate between the follicle and CL. The Doppler imaging of the uterine is like the Doppler imaging of the vaginal, but you have anatomically to differentiate between this cell. And we say this, for example, the applied anatomy. I have to know applied anatomy very well to know the blood vessels and then to describe the changes of in blood within these blood vessels.
I see. Well, thank you so much, Dr. Mohammed, for your intriguing presentation and the pivotal information that you shared that's going to improve the performance in animal reproduction. So I would like to thank the National University of Columbia and to Professor Linus as well. And we will see you all again in our 5th seminar, which will be on March 16th. And I hope everybody has a great rest of their...
Thank you very much, Amy, and thank Dr. Yasser very well, and all the people which helped me today in this presentation.
Bueno, no, dándole las gracias a Emi Montgomery de la Universidad de Missouri, al doctor Mohammed de la Universidad de Mansoura de Egipto. Definitivamente hoy tuvimos problemas de conexión de la red de Wi-Fi específicamente debido a la locación que tenemos de nuestros colaboradores, Estados Unidos, Colombia y Egipto. Sin embargo, yo quisiera agradecerle a todos los asistentes al primer encuentro académico internacional de Ciencias Básicas Biomédicas. Es un evento organizado por la Facultad de Ciencias Agropecuarias de la Universidad Nacional de Colombia de Palmira, y también realizar un anuncio que se hizo al principio en inglés, pero que me parece una buena oportunidad. Queremos anunciar que usted puede ser el siguiente conferencista de este primer encuentro académico internacional de Ciencias Básicas Biomédicas. Sí, lo único que usted necesita hacer es enviarnos al comité organizador un abstract o un resumen de su investigación. Evidentemente tiene que estar enfocado en ciencias básicas biomédicas, no importa la temática, y el comité evaluador va a elegir si usted es un estudiante de pregrado, anímese; si usted es un estudiante de maestría, anímese; y si usted es un estudiante de doctorado, anímese. Pero si usted es un profesor que nos está observando y quisiera participar en este importante encuentro académico, pues los invitamos a que nos envíen esos resúmenes sin límite de palabras, que nos expliquen en este resumen su investigación a nuestro email que está apareciendo en pantalla. Este email que ustedes están viendo, encuentro-intercbb@unal.edu.co. Y además invitarlos a que sigan participando de nuestras próximas conferencias, que a partir de ahora serán investigadores locales. Es decir, ya con hoy completamos nuestras primeras 4 conferencias internacionales y vamos a continuar dándole el espacio a muchos investigadores de nuestro país, Colombia, que con mucho orgullo nos representan no solamente en nuestro país, sino a nivel del mundo. Con esto entonces me despido, no antes sin invitarlos a nuestra próxima conferencia, que será en el mes de marzo. Como siempre lo hacemos, estaremos indicando precisamente dónde y cuándo nos veremos con antelación, y también responder algunas de las preguntas que algunas de las personas del chat tendrían. El certificado del evento solo se dará a las personas que hayan cumplido con el 80% de asistencia y que adicional tendrían que esperar a que el primer evento académico internacional culminara, el cual está proyectado para el mes de mayo del 2021. Así que los invitamos desde la Universidad Nacional de Colombia a que sigan conectados con nuestro evento, primer encuentro académico internacional de Ciencias Básicas Biomédicas, y nos vemos el próximo mes. Muchas gracias.